Hello, this is Claire Jo again. I hope you have all enjoyed the talk so far. Next coming up is our closing keynote speaker, Dr. John Zuris, Director of Editing Technologies at Editas Medicine. I'm sure many of you are already familiar with John or at least have heard of his name or read his papers. His expertise and influence have made a significant impact in the field, and we're honored to have him join our stage. But for those of you who are newer to the field, please allow me to race through his bio. John obtained his PhD in biochemistry with Dr. Patricia Jennings at UCSD, then the postdoctoral studies with Dr. David Liu at Harvard, where he showed that Cas9 could be delivered efficiently using LNPs while also contributing to the group's seminal base editing work. John then joined Editas and then has led the development of multiple gene editing platform advancements in the past 8 years. One example is the development of a proprietary engineered Cas12a nucleus, described in a 2021 Nature Communications article, which is used in both Editas sickle cell and beta thalassemia clinical programs. John also led the development of a new transgene insertion strategy called SLEEK, recently reported in Nature Biotechnology, which relies on a unique selection mechanism to drive knock-in edits over indel edits. John Scribb was lucky enough to be able to help contribute to development of SLEEK technology by supplying non-viral single-stranded and double-stranded DNA templates used for this project. And today here in the stage, he will be sharing with us his new exciting work in this talk titled SLEEK, a highly efficient transgene knock-in technology in clinically relevant cell types. Without further ado, John, please feel free to start your presentation.
Hello, everyone. My name is John Zuris, and I'm the Director of Editing Technologies at Editas Medicine. I would like to thank the conference organizers for giving me the opportunity to present my work here today. I will be speaking about an exciting technology called SLEEK that we have developed for highly efficient knock-in of transgenes in clinically important cell types, which we believe may advance the development of next-generation cell-based medicines. So here's my disclosure slide. So we have seen many examples of CRISPR-based gene editing in clinical trials, such as those for sickle cell disease and CAR-T therapy. Allogeneic CAR-T therapy is probably the best example of why efficient transgene knock-in is so important for generating cellular medicines. Despite early progress in the CRISPR field and demonstrations of highly efficient gene knockout, knock-in of functional transgene cargoes continues to remain a challenge. In ex vivo therapies, there's often high heterogeneity in engineered cells being administered back to the patients after gene editing, which can reduce efficacy and potentially pose safety issues. Our goal was to develop an editing technology that could fundamentally improve transgene knock-in and the generation of cellular medicines. So CRISPR gene editing is, of course, our core technology at Editas Medicine. We had to first decide which nuclease we wanted to use from a suite of nucleases available. We were building out our specificity platform at Editas, and we observed that SP-Cas9 is not a very specific nuclease, similar to what many others have also reported. We observed that SP-Cas9 is prone to editing at potentially hundreds of sites in the genome. So in this digenome specificity experiment, we chose matched sites in the human genome that could be targeted by both SP-Cas9 and AsCas12a. As you can see, AsCas12a is 10 to 100 times more specific across the same locations in the genome that we examined in this study. In fact, the majority of AsCas12a guides hit only one intended on-target site. However, when we first started working with AsCas12a, we found that it was less efficient than SP-Cas9 in general across target sites. Importantly, this issue was overcome a few years ago through a collaboration with IDT, with the resulting work jointly published by our groups in 2021. Together, we showed that AsCas12a can be engineered to be almost 100-fold more potent than the wild-type version of AsCas12a, which is shown here in T cells on the left. When we tested the engineered AsCas12a on multiple target sites in the B2M locus, we found that it could edit all of the sites with similar potency, which is shown on the graph on the right. Here, each black dot represents a different guide. In our manuscript published in Nature Communications in 2021, we observed this same trend across other clinically relevant cell types, including HSCs, iPSCs, and NK cells. Now, importantly, this nuclease was able to retain its high intrinsic specificity that is normally seen in the wild-type nuclease. So, despite major progress made in the gene field, efficient knock-in remains a challenge. Here you can see that we can achieve close to 100% knockout of 4 key T-cell targets simultaneously in a single experiment. However, we achieve 60% knock-in efficiency for a single transgene cargo delivered using an AAV6 donor template. A cell therapy generated in a similar manner would include many cells that do not contain the intended beneficial edits, particularly the knock-in cargos. So here we propose a solution to the knock-in challenge called SLEEK, which stands for Selection by Essential Gene Exon Knock-in. SLEEK is a knock-in selection method that uses our engineered AsCas12a, delivered as an RNP, along with an HDR donor template. The AsCas12a RNP and DNA donor template are electroporated into our cells of interest, and a combination of indels and HDR repair occurs in our population of cells. SLEEK is based on the ability to drive HDR or knock-in repair over the more common indel-forming NHEJ-mediated repair through a selection process that's created by targeting essential genes such as GAPDH, where indels will result in cell death. Because our engineered AsCas12a achieves nearly 100% indel formation, as I showed you on the previous slide, we hypothesized that we could essentially remove the unedited cell population from the list of expected outcomes below. Because of the negative selection for cells that receive an indel that do not receive correct insertion of the transgene cargo, the predominant cells that show up after 1 week of expansion and culture should only be those which received correct HDR-mediated knock-in of the transgene cargo. Furthermore, by targeting an essential housekeeping gene with very high endogenous expression, we can ensure that our cargo is highly expressed. And this avoids silencing over time, which is of particular importance with iPSC-derived cell therapies, which must go through a long differentiation process before becoming a mature cell. So here is a diagram of our SLEEK knock-in construct design. We are targeting the last exon of the GAPDH gene. Our knock-in construct includes a restored version of the remainder of the exon downstream of the cut site, which is highly recoded to prevent any recutting after repair and to avoid being accidentally used as a 3' homology arm during HDR. We then insert 1 or more transgene cargos of interest separated by the P2A ribosomal skipping sequence. This is followed by the stop codon and all of the other components required to make a functional mRNA. After translation, we should have a functional GAPDH protein and fully functional transgene proteins. So we first tested SLEEK in iPSCs and demonstrated reduction to practice. We performed a knock-in time course experiment to show the enrichment of SLEEK GFP knock-in cells taking place over time. As you can see in these flow plots, the percent of cells expressing GFP goes from 11% to 93% over a course of 9 days after electroporation, confirming that there is a selection of knock-in edits over knockout edits. Importantly, for this low-level initial plasmid knock-in to enrich to over 90%, we must have had very few unedited cells during the initial electroporation stage of this experiment, and we believe that our highly efficient engineered AsCas12a was critical for achieving this outcome. So here we show that SLEEK is a highly versatile method as it can work with several different donor types. This is shown on the graph on the left where we compare several different non-viral donor templates in iPSCs, including those offered by GenScript for GFP knock-in. We observed greater than 90% GFP knock-in irrespective of the donor format. Furthermore, the graph on the right shows that by using SLEEK at other essential genes, like TATA binding protein or KIF11, for example, we could tune expression of our transgene cargo as the levels of GFP expression are different depending on the essential gene and the essential gene promoter. Hence, SLEEK allows for tunability of cargo expression because of different endogenous promoter strengths at different essential genes. So here we show that we can use SLEEK to insert a 5 kilobase knock-in cassette with 4 different genetic cargoes. So in this case, we have CD16, IL-15, which are both important for iPSC-derived NK cells, and then also 2 additional cargoes that are easy to detect in GFP and M-cherry. We validated the expression of all the detectable cargoes in iPSCs by flow cytometry. We observed greater than 90% knock-in efficiency for each of them except CD16, which cannot be detected at the iPSC stage as it requires adapter proteins that are present in mature immune cells. An important thing to note is that we observed that the immune fluorescent intensity, or MFI, of the GFP as the 3rd cargo in this 4-cargo construct was about 40% fold lower than when the GFP was the only cargo in the cassette. This indicates that we need to select cargo position carefully in a multi-cistronic construct with cargoes requiring higher expression positioned at the 5' end and those with lower expression desired to be positioned more at the 3' end of the construct. So having shown that SLEEK works well in iPSCs, we wanted to check if it works in other clinically relevant cell types. So here we show that SLEEK enables us to achieve the best-in-class knock-in efficiencies observed to date, to our knowledge, in T cells, B cells, and healthy donor NK cells. Importantly, we tested SLEEK against the gold standard track knock-in strategy used to knock in cargoes into T cells. As you can see, our SLEEK process not only is more efficient than track knock-in overall, but is an order of magnitude more potent, which means that we could use far less AAV6 per treatment. In addition, we did not see any notable negative effects on cell viability or expansion after 7 days compared to the track knock-in strategy or the mock treatment. So our ability to achieve close to 100% knock-in in T cells enables us to pursue our goal, which is to generate highly homogeneous gene-edited cell-based medicines. Here we are showing potential editing strategies that would likely be needed to achieve beneficial clinical outcomes with a T cell or NK cell oncology medicine. A critical step for CAR T therapy is to have efficient insertion and expression of the CAR. The graph in the middle shows that we can achieve over 95% knock-in of a CD19 CAR with SLEEK in T cells. These CD19 CAR SLEEK knock-in T cells were then put into a tumor-killing assay in vitro and showed robust cytolysis against tumor cells that express CD19. Next, we show that SLEEK can potentially be used to generate allogeneic T cell medicines, which require many edits. The graph in the middle shows successful knockout of the TCR and MHC class I coupled with SLEEK knock-in of a CD19 CAR. These cells have nearly 90% of all the desired edits as measured by flow cytometry. These CD19 CAR SLEEK knock-in T cells were then put into a tumor-killing assay in vitro and showed robust cytolysis against tumor cells that express CD19. So in addition to the B2M and C2TA knockouts for inhibition of host CD8 positive and CD4 positive T cell responses, a third key component of an allogeneic T cell is host NK cell inhibition. Here we show successful knock-in of an NK shield in primary T cells using SLEEK. We achieved 93% knock-in of a B2M HLA-E fusion construct in primary T cells, which has been shown to inhibit NK cell responses in several formats. SLEEK NK shield knock-in T cells were tested in an NK cell degranulation assay to check for NK cell activation and degranulation using CD107A as a marker. As expected, T cells with the B2M knockout only induce an NK cell degranulation response as compared to unedited cells, as shown in the graph on the right. Importantly, the SLEEK knock-in of an NK shield reduces CD107A degranulation response levels to those similar to the unedited cells. This indicates SLEEK knock-in of a NK shield into primary T cells offers protection for our cells. Now, to make multiple knock-in edits, such as a CAR and an NK shield, would presumably require multiple AAVs, which poses manufacturing challenges, and AAVs have packaging limitations as well. Non-viral DNA templates are less efficient at knock-in than AAV6, unfortunately. We reason, though, that because SLEEK has a selection mechanism, we could still achieve high knock-in efficiency with these non-viral formats. So here we show that SLEEK works with several non-viral DNA templates in primary T cells, achieving similar knock-in rates to the AAV6 gold standard. And again, I wanted to highlight that some of these different constructs here came from the GenScript. We found through optimization that we could match the AAV gold standard on efficiency, viability, and expansion. For simplicity, I'm highlighting just two non-viral donor template formats and comparing to AAV6 and the mock treatment. Showing this allows us to generate by doing this, we can show that we can generate larger knock-in cassettes containing multiple transgenes by avoiding the AAV6 cargo capacity issue. Next, we want to demonstrate that knock-in of functional cargoes with non-viral DNA templates could be achieved. Here we show knock-in of a C19 CAR or an EGFR CAR into T cells with high efficiency in the top panel. And applications of this could be, for example, for autologous CAR T or engineered TCRs. The bottom panel shows that we can knock in a bicistronic construct expressing both a C19 CAR and a shield with high efficiency, importantly using a non-viral DNA template. Next, we show an example of knocking in a CAR into healthy donor NK cells. We then tested the CD19 CAR SLEEK knock-in NK cells in a tumor-killing assay in vitro and show robust lysis of tumor cells that are positive for CD19. Because achieving efficient knock-in is widely recognized as challenging in NK cells, we believe SLEEK will enable better engineered healthy donor NK cell medicines. Lastly, I will show you how SLEEK enables us to generate iPSC-derived NK cells, or iNKs, to target tumors in vivo. So here we are leveraging SLEEK to generate a highly engineered iPSC-derived NK cell with enhanced tumor-killing function persistence. So natural killer cells are strong effectors of the innate immune system, and can recognize and kill tumor cells, making them attractive for use as cellular cancer therapies. NK cell effectiveness, however, is diminished by the lack of persistence due to low levels of critical cytokines required for NK cell survival, such as interleukin-15 or IL-15 for short. NK cells naturally express CD16, which is recognized as antibodies that bind tumor antigens, thus promoting antibody-dependent cellular cytotoxicity, or ADCC. Their ability, though, to exert ADCC is impaired when CD16 is cleaved off after NK cell activation. Here we are knocking in a membrane-bound IL-15 complex and CD16 as a bicistronic cassette into iPSCs using SLEEK. The resulting differentiated iNKs should have enhanced ADCC due to constitutive overexpression of CD16 through the CD16 knock-in, and enhanced survival and killing due to increased IL-15 signaling through the knock-in of the membrane-bound IL-15 complex. So here we show that our SLEEK double knock-in iNK cells do indeed have improved tumor killing and persistence in vitro. On the left graph, you can see that our SLEEK double knock-in iNKs show much stronger surface expression of both CD16 and IL-15 receptor compared to unedited iNKs. In the center graph, you can see that SLEEK double knock-in NK shows superior tumor killing in a 3D spheroid tumor killing assay, both with and without trastuzumab, which is an antibody which allows them to perform ADCC. And this is due, we believe, to the higher levels of CD16 through the SLEEK knock-in. And finally, on the right, you can see that our SLEEK double knock-in iNKs show improved persistence in culture without supplying additional IL-15 cytokine, which we attribute to high expression of membrane-bound IL-15. So we evaluated the anti-tumor activity of SLEEK double knock-in iNKs in a SCO3 tumor-bearing mouse model. Mice were injected with unedited or sleek double knock-in iNK cells intraperitoneally with trastuzumab, as noted on the upper left panel. We observed strong tumor reduction out to 112 days, which is shown in the upper right figure. Our sleek double knock-in iNK shows strong tumor clearance in the presence of trastuzumab, demonstrating the importance of high CD16 expression, as shown on the bottom left by IVUS imaging. And importantly, on the bottom right, the survival curves show impressive survival for mice receiving the sleek double knock-in iNKs with trastuzumab compared to the iNKs that received trastuzumab alone. We believe this strong and durable tumor killing in vivo is due to high levels of CD16 and IL-15 expression because of our sleek double knock-in. So lastly, we show that sleek iNKs were able to persist after 144 days post-dosing without any exogenous cytokine support, indicating the importance of tying expression of our two functional cargoes, in particular the membrane-bound IL-15, to the strong GAPDH promoter through targeted knock-in using our sleek technology. So in summary, our goal was to develop an editing technology that fundamentally improves the generation of cell-based medicines. We see broad applications of sleek in many cell types, and we hope to leverage this technology for a wide variety of applications with current and future partners. In addition, I would like to refer you to our recent publication in Nature Biotechnology, which covers all the work I presented today in greater detail and includes construct sequences and critical protocols to ensure successful usage of sleek. And with that, I thank you all for your attention. I'm happy to take any questions.
Thank you, John, for the great talk. I think we will now start our live Q&A session. So for the audience, if you haven't submitted your question yet, please do so in the question box you see below in the screen. And I'd like to first comment that the efficiency I think you show here with the sleek technology is amazing, especially with the non-viral approaches, where I think most researchers are kind of struggling to obtain 40, 50% knock-in, where we're showing 80, 90% or even higher. And I think since the publication, you must be getting tons of questions and tensions because of this great work, as even as GenScript said, we get quite a couple requests to repeat. So congrats on the great work you've shown here. And I guess my first question is, Dr. Wagner this morning has, mentioned your work. I wonder if there's anything you want to comment in response to that.
Oh yeah, sure. Yeah, one of the things I think that's real important to acknowledge is that, if you look at the presentation in the manuscript, we start out with AAV6 because that's sort of the gold standard. You can already get real high rates, without necessarily a technology like sleek there. But the real value, as you noted, I think is going to be in the non-viral, templates that you can use. And actually, in order to pull that off, there's a lot of key work that was done actually by Demetrius Wagner's group, also, Alex Marson's group, and also Brandon Moriarty. I wanted to call out those three groups because one of the things that if you've read their papers from those groups, you'll see is that when you're delivering, for example, double-stranded DNA, and Demetrius mentioned this earlier, but that is not something T cells want to see in their cytoplasm. And so there's a lot of key ways to sort of circumvent that issue, including, inhibitors of like, the C-gas, for example. But the one thing we actually really like to use, and we highlight this in the methods in the sleek manuscript, is actually the criticality of editing these cells, about 36 hours after they come out of thaw, because it's actually during that time they really haven't turned on those pathways, and others have, showed that. So that's something we leverage with sleek. The other piece to that as well is I highly encourage people to use these sort of HDR enhancers or sort of NHEJ inhibitors, especially if they're new users of non-viral vectors, for example, because this will actually give you more of a margin for error. If you're a very seasoned user, you don't actually need these things, as we show in the manuscript, for sleek to work. But starting with that's probably a good idea.
Great. And I think typically, as you know, the Marson lab, even Wagner that we work with, typically we see single-strand DNA tends to be performed better in terms of efficiency and toxicity. But with the sleek technology, it seems like both viral, non-viral, everything is pretty comparable overall. So do you have any comment on that? If, for example, moving forward into clinical applications, what is your recommendation in terms of selection of different payloads?
Yeah, so I think between clinical and research, there's different pros and cons to using these different template types. I think, I mean, the real beauty with Sleek is that you could use, for example, a circular plasmid actually to get 90% knock-in, let's say. You know, in iPSCs, about 80% in T cells. That's great because that's going to be the easiest thing to manufacture GMP. We already can do this. You know, so that I think will be a great route forward for clinical applications potentially. I think if you are trying to get the maximal efficiency, I do think the single-strand DNA is going to be very good there. It's probably also going to be tolerated the best by these cells. And one of the things to get around some of the toxicity of double-stranded DNA, with Sleek, the key there is you get the selection of the knock-in, so you can actually use lower doses of double-stranded DNA templates, so it limits toxicity. The good thing is with single-strand DNA, that's already less toxic, as you noted. So you can potentially use higher doses, get higher efficiency, and actually for folks doing clinical applications, though, that's actually important because that actually determines how many cells are going to survive after that editing event, and then that whole expansion that kicks off. You need to get to a certain cell number at a certain date in order to get that back to someone or in order to produce enough of your product. So big fan of single-strand DNA. And I know you guys, for example, have made a lot of progress in increasing the length at which you can design this, because I know one thing people might want to do here is put in multi cargo cassettes. And of course, that's easier right now with double-stranded DNA.
Sure, and thanks for the comment. And I think to follow up on the questions from the audience coming in, what are the in vivo applications where Sleek approach would be appreciated?
Well, yeah, I think everywhere that Sleek can work, not necessarily, it's not anything new from where we already would want to use a knock-in. So of course, CAR T would be an exciting place for that. What we show in the paper, and this is actually something we intend, our partners intend to go clinical with, is actually a iPSC-derived NK cell that actually uses Sleek to knock in the cargo. Now, you don't have to necessarily use Sleek in the context of an iPSC because of the fact that you can pick a single clone. But what Sleek allows you to do is leverage that with multiplex editing and actually still be able to find that clone that is free of translocations, no off targets, has all your knockouts, including frame shift mutations with your knockouts, and has your knock-in. So Sleek really does enable iPSC-based medicines, I think, as well, which is really exciting. The other thing, too, is NK cells are obviously a lot harder to work with than T cells. So Sleek is probably going to be very useful as a way to potentially go non-viral for NK cell editing. That's just a much taller order than I think T cell or iPSC. And so, having a selection mechanism like that, I think would be very valuable. Yeah, I think there's also a key part of this is in the context of developing a therapeutic in the research stage, you want to be able to test lots of things very quickly. And Sleek, I think, will allow you to test lots of different configurations of, let's say, a CAR or multi combinations of different, let's say, cytokines and CARs and, let's say, NK inhibitor shields that you may want to test in your product. You can more easily do all this without having to go make an AAV6 vector to actually do your research, in your company or in your lab. And so that's one of the other pieces. Hopefully this will accelerate, people's ability to create products they want to take forward into the clinic.
Great, and I think to follow up on the multiplex knock-in, so one question is, do you see differences when doing multiplex knock-in between the knock-in, I guess, size or versus a single knock-in with a more polycistronic design?
Yeah, that's a great question. So My recommendation, if possible, is if you want to put in like a 2 or 3 cargo cassette into your cell, be it an iPSC, T-cell, NK cell, just knock it all into the GAPDH site, which is what we focus on. You're going to get great knock-in efficiencies. It's been the most robustly characterized. And then on top of that, you get really high expression, which almost in all cases is what you want with your cargo. There'll be exceptions to that. But I would encourage you just to make multi-cistronic cargos for that site. I also think, you know, again, when you're making multiple knockouts and knock-ins, if you're thinking clinically, you're also thinking about having to make those constructs. And if you can put all your cargos in tandem, that's just one GMP plasmid or single-strand DNA or whatever you choose. That's just one of those that you actually have to make and characterize. And I think that would be advantageous there.
Great. And one other question is, you highlighted that no exogenous promoter is needed, but would adding one be helpful?
That's actually a great question. So there's a couple things you can do there. One of the things we show in the supporting information, the paper, is that we can knock in 4 cargos in tandem. And cargos 3 and also importantly cargo 4 still express, right? Because your question is, do those expressions fall off a cliff? Well, those expression levels do drop though from if those cargos were in position 1, like in the construct. And so for that reason, one thing you could consider is if you especially if you don't have really a cargo limitation, you know, or I mean a size limitation if we're talking about non-viral, right? Then you could consider actually dropping new promoters actually in those regions. And that's one way you could potentially address that if you really wanted the maximum expression for all of these cargos. That's an area that's not fully explored yet. You know, and so I actually encourage researchers out there to kind of, you know, let's see what else can be discovered there, because I think there's a lot more that can be done in that realm as well.
Great. I think we have a couple more questions here, but we'll probably take 2 more. So one is the improved persistence of your CD16 MBL IL-15 CAR T cells is impressive. Are there any downfalls to having the injected cells persist beyond the presence of the tumor?
There could be. So just one thing I want to note there is that that's in the context of an iPSC-derived NK, not a CAR T cell. But yes, so that the data shown there, and that's in figure 5 plus some of the support information in the manuscript, but the data shown there, it's very clear that not only is SLEEK useful for knocking in cargos, but it's that GAPDH promoter was critical to actually getting the expression levels you want to get the persistence there of the NK cells. And NK cells are not great at persisting. So there, the hypothesis and the technology we have merged together in a real beautiful way. If you're talking about CAR T, it's quite possible there's certainly data out there that suggests in some cases you may not want your CAR expression to be quite as high, or there could be other cargos you have in tandem, you know, like maybe an IL-12 or something. You may not want that to be as highly expressed, and so that's an example of you know, why you might want to knock into another site, you know, like tata-binding protein or KIF11, which you can see have, you know, literally orders of magnitude lower expression levels. It's also a reason you could consider putting in like a promoter that maybe has lower expression.
Great. And I think just one last question is, do we have any plans to commercialize SLEEK? Are there any possibility to license this technology that you guys are looking at?
Yeah, so as just a general statement, I mean, this is a technology that we think is very powerful. This is something we want to, you know, help as many patients as possible, potentially. And so this is why we're very eager to partner around this technology. As you can imagine, there's a lot that can be done here. There's so many applications in the iPSC space to make all sorts of cell types from that. There's obviously the T cell and the NK cells, and then B cells. You know, we show examples of that there. So all of these places, macrophages, others, these are all places where, you know, I think we'd be happy to partner potentially with people. And as always, feel free to reach out. You have my contact information and email. So yeah, encourage all those. opportunity.
Thank you. And thank you, John, for the great talk. And thank you for all the audience to attend. And I hope you have enjoyed today's talk at the GC Summit. And it will be actually our award ceremony coming up right now. So please do join at our next stage and celebrate with us. Bye. Thank you.
Thank you.